Welcome to the Molecular Therapy Podcast. I'm Dr. Rory Bricker Anthony, Scientific Editor of the Molecular Therapy family of journals. This episode features a conversation between Dr. Paloma Giangrande, Editor-in-Chief of Molecular Therapy Nucleic Acids and Chief Technology Officer at 11 Therapeutics, and Dr. Robert Bell, Chief Scientific Officer, and Dr. Michael Ehlers, President and Chief Executive Officers, both from Ascidian Therapeutics. They will discuss a recent article published in Molecular Therapy Nucleic Acids by Dr. Bell and colleagues titled RNA Exon Editing, Splicing the Way to Treat Human Diseases. Did you know that this is the final episode of the Molecular Therapy Podcast before the close of ASGCT's 2025 annual meeting call for abstracts? That's right. Abstract submission for the 2025 annual meeting in New Orleans closes at 11:59 p.m. Eastern Time on Friday, January 31st. Don't miss your chance to present your work at the premier event in cell and gene therapy and have your abstract published in the Molecular Therapy Supplement. Find submission guidelines, important dates, awards and incentives, and so much more at annualmeeting.asgct.org/abstracts. And now, Dr. Giangrande, Bell, and Ehlers.
Mike and Bob, thank you for being here with us today. And thank you for your great paper that was recently published in our journal. Let's dive right in with an introduction to RNA Exon Editing. Who wants to take this?
Bob, you want to start?
Yeah, sure. And thanks, Paloma, for having us. It's a real pleasure to be able to do this today. So RNA Exon Editing is an approach that we've been developing at Ascidian that leverages somewhat ancient biology, and that's known as RNA trans-splicing. So at a really fundamental level, it's the concept by which two distinct pre-messenger RNA molecules can be spliced together, right? So typically when we think about splicing in RNA processing, it's splicing in cis, one pre-messenger RNA, the introns are removed, exons are joined. Well, what happens in nature is that you can splice in trans, two separate molecules coming together. And we've been leveraging this for therapeutic purposes here at Ascidian Therapeutics.
And so RNA trans-splicing has been established in mammals for many decades. If I'm not mistaken, back in 1980s or so. Why now? Why has it taken so long to take RNA trans-splicing into the clinic?
Well, maybe I can start and then Mike, you can add. I think one of the things that has been just so exciting in the field of genetic medicine, gene therapy, RNA basic biology is just we now have the tools that we didn't have even 20 years ago. So for example, large synthetic DNA library, barcode screening approaches, next generation sequencing combined with, bioinformatic computational science allows us to do things and to set up screens to identify molecules that have much greater efficiency and much greater selectivity. And then you combine that with just our ever-growing knowledge of how RNA splicing works, how RNA structure impacts RNA function in splicing. And so I think, why now is because we can combine all of the latest and greatest synthetic biology tools that exist with the knowledge of how splicing works to really turn these into therapeutic molecules.
Yeah, that's great. We've been hearing a lot about many different types of editing approaches recently, including genome editing, RNA mediated or ADAR mediated RNA editing. Can you go into how RNA exon editing differs from some of these other mechanisms of editing?
Well, I can take that one. I suppose we can trade off here a bit. Yeah, I think there are several unique features about RNA exon editing that allow it to do some things that you'd be challenged to do with other approaches. For one, we can edit thousands of bases at a time at the RNA level. So our lead program, which is currently in clinical development, I think we'll have a chance to talk about that. We replace over 3,700 nucleotides of RNA in a single reaction. So that's a scale at the kilobase scale that I think is quite different from a number of the editing approaches. Second key feature is that we co-opt endogenous cellular machinery, so we don't need to express a foreign enzyme, such as a Cas nuclease or a reverse transcriptase, dead Cas fusion, or some of these other methods that are required to mediate editing. We simply co-opt endogenous machinery, so we don't need a foreign enzyme to be able to do that. I think one key thing to reemphasize here that I think is implicit, but I'll just reemphasize it, is that this is editing RNA and not DNA. So that comes with, a degree of differences, but also avoids some of the liabilities that one might have on permanent genome editing, if you will. So those, I think, are some of the key differences. Now, we always think that these different RNA therapeutic modalities or editing modalities will have a place depending on the specifics of any given genetic change that you want to make or any specific disease entity. But in the case of RNA exon editing, we do think that there are a number of diseases and disease targets for which this technology will be uniquely advantaged.
That's a great transition to the next question. Mike, do you want to highlight some of these indications or mechanisms that you were alluding to?
Yeah, well, there are lots of things you can imagine doing if you can replace thousands of nucleotides of an RNA molecule at once. I'll highlight two types of applications, but certainly not necessarily limited to these types of applications. One of the first, and perhaps one of the more obvious ones, is where you've got a genetic disease, where you've got a very large gene with high mutational diversity, which is to say a large gene for which standard single vector gene replacement by AAV, for example, would not be feasible because the gene is simply too large. And those large genes where many different mutations can cause disease, where a base editing approach would require dozens or hundreds or sometimes thousands of different agents to be able to address the full range of patients and patient mutations. So large genes, high mutational diversity across the gene are particularly good targets for us. And that's exemplified in our lead program, by the way, which is for a gene called ABCA4 in the most common inherited form of blindness called Stargardt disease. And maybe we'll come back to that, I think. The second category of targets or diseases that we think are particularly amenable are those diseases where the genetic cause of the disease is a mutation that can be, say, a toxic gain of function type of mutation. And where we know with RNA exon editing that you can, in one reaction, get rid of that toxic mutation and replace it with wild type. So you get around a lot of the questions that can hamper those areas, like, is it really gain of function? Is there a degree of haploinsufficiency? Is there an endogenous function of the gene? Could you, where it's a risk if you knock down both the mutated and the non-mutated? RNA exon editing allows you to essentially skip all those questions and simply, in one reaction, replace a potentially toxic allelic transcript with wild type. So those are two examples. I'm sure there are others that we could use, but we've been focusing on those across our pipeline.
Yeah, and actually that leads into another question that I had for you guys, in particular, how RNA exon editing compares to other RNA-based therapies that are already in the clinic, such as RNA interference or RNAi, as well as antisense oligonucleotides that are really going after some of these gain of function, toxic gain of function type of indications that you've alluded to, or even therapies like mRNA therapy that are going after loss of function type of diseases. Do you see RNA exon editing as complementary to many of these other approaches, or it seems like you alluded to the fact that RNA exon editing can now do both of these things in one pot, essentially, or kill two birds with one stone, essentially. I just would like to hear your thoughts on that.
Yeah, I mean, I think as Mike mentioned, for those targets where you may not only want to knock down, but to restore wild type protein, I think RNA exon editing has advantages. Or if, you're taking an oligonucleotide approach to, do some type of very specific exon skipping or, to eliminate a specific variant, whereas we can restore wild type. The other aspect of whether it's mRNA gene replacement or even just conventional AAV gene replacement, one of the things that I think has been a challenge in the field is maintaining the right level of expression of whatever you're trying to deliver. And that means expression in the target cell types or preventing expression in non-target cell types, right? There are examples where overexpressing a protein in the wrong cell type at the wrong level can be extremely toxic. That's, we know that based on, duplication syndromes for certain targets, but we've also seen it just in conventional AAV gene therapy throughout the years. One of the unique advantages of exon editing by transplicing is that the cell controls how much pre-messenger RNA transcript is present, right? So there's an inherent sort of selectivity in the sense of if a cell is not normally expressing that pre-messenger RNA transcript, the exon editor doesn't have anything to edit, and you're no longer at risk of expressing a protein at the wrong levels or in the wrong cell types. So I think we view that as being a really big advantage to targeting pre-messenger RNA and allowing the cell to participate in control of the therapeutic expression that you achieve.
For those of us who have taken our biology, intro 101 to biology decades ago, can you say a little bit more about transplicing versus cis-splicing, and if transplicing reactions occur in direct competition with cis-splicing? And if that's the case, how do you ensure that your RNA exon editors favor transplicing reaction?
Yeah, great question. So Mike, do you want me to start with this one?
Yeah.
So, there's a couple key, if you will, rate limiting steps. But to answer your question on the short end, yes, we are in competition with cis splicing. So at a fundamental level, we are outcompeting splicing and cis. And the way we can do that is a first critical step is identifying a location on the target pre-messenger RNA, an intronic region that we can bind to. And that, again, is where we can now leverage, whether it be pool-based NGS screening. Some of the introns you may be interested in target are tens of thousands of kilobases. So finding an ideal location to engage with is a critical step in ensuring you can outcompete. Ascidian has developed a number of tools, empirical experimental workflows, as well as in silico prediction models that allow us to identify optimal intronic targeting sequences. The second piece is, again, getting back to our knowledge of how splicing works. We can now incorporate elements into our exon editors. And these are human-based sequences that have known RNA protein binding functions related to splicing, so that when our exon editor is engaged with the target, the splicing is preferred in trans as opposed to in cis. And so those are just two of the flavors of things. And as you can imagine, we've identified a number of other strategies to, for example, boost stability of editors, to incorporate mechanisms to drive location of the editors in terms of nuclear subregions. And all of that together allows for the type of editing efficiencies that we've been able to achieve.
I would just add is that an interesting thing is there's a lot of basic biology that we still don't understand, that we'd like to understand about trans-splicing, and it's undoubtedly going to be a complex interplay between a lot of the things that Bob was just talking about there, which we know that splicing reactions occur co-transcriptionally. We know that different exons and introns undergo different kinetics of splicing. We know that there's a whole host of different and dynamic RNA binding proteins that participate in the process. And so there's a lot of very basic biology that we really just don't know. So I'm hopeful that not just at Ascidian, but that the work that we're doing at Ascidian sparks a lot of interest amongst the academic community to be able to understand this more. I think that we'd like to understand much more about the structural biology, the cell biological dynamics of this, and be able to use this to engineer our RNA exon editors, which already require a fair bit of sophistication in engineering multi-modular RNA molecules, and not just in the modules they have, but in the codon optimization and other certain features. Bob mentioned sequences that recruit elements of the splicing machinery, but there's a lot more to be understood at the basic level.
Yeah, and that's great. So, so in addition to some of these, I guess, molecular understandings of trans-splicing versus cis-splicing, and trying to compete with these endogenous molecular mechanisms, what are some other technical challenges that you have faced with the RNA exon editing approach? And maybe, governing delivery, as you also alluded to, subcellular localization, the proper subcellular localization, and how are you going about addressing some of these other technical challenges?
Well, I think one is really not necessarily specific to RNA exon editing, but it's worth discussing, and it's delivery. When we initially set up Ascidian, it was on the premise that we want to be able to use validated delivery strategies. We really view ourselves as a company focused on payload innovation. And so, part of the reason why we've started with our first clinical program using in the eye, where we're using a validated delivery, we're using a natural occurring AAV serotype. But, there are a lot of applications where biodistribution is challenging, and we're very excited about the emerging sort of next generation vectors and even some non-viral delivery technologies. Especially, for example, you could imagine applications of RNA transplicing where you want to target cells that are continually undergoing division, right? So non-terminally differentiated cell types. And for that, right, an AAV vector wouldn't be appropriate. So I think that's something that we think about all the time in terms of our pipeline prioritization and the programs we're going after is really ensuring that we can build the editors to target a number of genes, but just really making sure you have that sort of translational path to delivering to the right cell types in the clinic.
And yeah, and that's great. So it seems that your technology is very much agnostic to the, to the delivery approach that you can use, and you can easily use a viral vector as well as an LNP nanoparticle that, again, de-risked approaches to delivery that can also enable RNA exon editing. And so earlier you had touched a little bit on how you design these RNA exon editors to identify their endogenous target, their pre-mRNA within the cell. Can you discuss a little bit about the potential off-target effects that you could encounter if this targeting mechanism that you're engineering is not quite, perfect? And how do you go about working to minimize potential off-target effects?
Yeah, well, great question. I'd say the first answer is we start assessing that very early in a program's life cycle. And in fact, use it to help us, actually optimize and screen. And the way we approach it is a couple complementary methods that I can talk about. One, for example, in initial discovery. Well, first I'll say this. We truly do an unbiased approach. A lot of gene editing technologies will start off by in silico prediction of something like a guide RNA homology, and then just go and assess whether or not those targets are altered. That has not been Ascidian's approach. We're truly taking an unbiased approach, looking at whole transcriptome level changes. And so, for example, in discovery, we apply things like PCR amplification to really develop very sensitive methods to identify any type of off-target transcript. We've leveraged emerging, long read sequencing as well as hybrid capture type of technology to validate whether or not there are any real meaningful off-target transcripts. We look just at very deep whole transcriptome sequencing, things for intron retention profiles to make sure that we're not having issues in that respect. And, what I can say is as it's part of our workflow, we screen and we select molecules that have a very selective profile, and then we triage molecules that maybe are less selective. What I can say about, for example, the lead program, ACDN01, the one thing that was quite surprising to us, but fortunate to some extent, is that the degree of off-target editing that we've been able to detect with those sensitive methods is orders of magnitude, in some cases 1,000-fold lower than normal errors that happen with things like transcriptional read-through or other splicing errors within a cell. And while we can detect a very small amount of off-target editing, you then can follow up and ensure that it's not creating any sort of immunogenic protein or some type of. There are no examples where you're changing the total level of that transcript, right? And of course, through a number of tox studies, safety studies, you gain confidence in the low risk of any low-level off-target editing with transplicing. And I think the most important thing, especially from like a regulatory perspective, is we've done those assessments in multiple biological systems. We've done them in various cell line systems, discovery screening. We've done that assessment in our safety studies in non-human primates, and we've done a lot of that work in human retinal explants for the lead program, which is, again, a retinal targeting therapy. And again, you look at the totality of that data to really gain confidence in the risk profile. Somewhat controversial. I think when I hear of gene editing technologies going out and saying they have 0 off-target edits, in my opinion, they're probably not using
It's
the most
hard to believe.
technologies to identify them. And I think what we should be doing as drug developers and working with the agency is taking advantage of the most sensitive technologies and evaluating risk, and not just setting thresholds that we see no off-target editing. Mike, I don't know if you have anything to add on there.
I think that was very thorough. I mean, at the core of it is not just our methodology, which Bob walked through in a way, but the fact that this is an RNA editing technology as well, which always means that it's not really a permanent stamp, if you will, and it's a fraction of the transcripts in any given gene. And that's very different from a DNA edit, for example, and the corresponding risk. And we've seen that, and this is not just our view, but with our lead clinical program that's now in clinical testing, we had extensive dialogue with FDA on this very topic, amongst others, and they clearly understood both the data that we provided and I'd say the rigor of the approach that we utilized. And that was indicated by the extent to which we have to follow up on the patients that are part of the trial, which will be a 5-year follow-up. And for a lot of the genome editing things, the requirement is a 15-year follow-up. So I think that, along with the relatively smooth or very smooth, I'd say, positive interaction with FDA, I think highlights both the approach that we've taken and the fact that that we have very good selectivity and low risk, relatively speaking, on an off target.
And so you had mentioned at the beginning that although this is an RNA editing mechanism, so you're actually really going after the RNA and not modifying or editing the genome, but you're delivering a DNA vector that expresses your exon editor, if you will. Any concerns or anything that you've heard by the FDA, or in terms of potential for having to look at the whole genome effects of your RNA editor in terms of off-target effects? Has that been a requirement that they've asked for, or have you been able to satisfy regulators by just performing extensive whole transcriptome editing like you have?
Well, I'll start. I mean, FDA obviously cleared us to the IND for us to conduct the stellar trial that we're conducting with ACDN01 and Stargardt's disease, which we can talk about. They fundamentally, for purposes, and I'd say this is generally true across editing technologies, there is a desire to understand the degree of selectivity, whether there's any particularly common off-target effects in a way. We certainly don't see that. But the bottom line is usually the toxicology data that you generate across small animals, large animals, the safety data that you get in the clinic. I mean, that ultimately ends up being the bottom line for
absolutely
regulators and for what we're doing. I'd say the off-target effects, to the extent there are any, might give you a read into the sort of things to look at, but the bottom line is the safety profile in your toxicology and your clinical safety.
Can you share any insights from your recent clinical trial?
Well, what I'd say is that it's ongoing. We're enrolling and dosing patients, and we have developed a policy where we're not going to be talking about the data coming out of that until we've got a really meaningful amount of data we're really discussing, rather than anecdotal patient by patient sort of story.
Yeah, no, that makes complete sense. Can you discuss the current pipeline at Ascidian Therapeutics and diseases? I think you went a little bit into sort of the big picture of that, but diseases that you're targeting with your RNA exon editing technology.
Yeah, I think broadly speaking, it's in areas where, again, we have confidence in delivery approaches have been solved and there's clinical precedent, or there are a few, I'd say, in our pipeline where we think it's close and we're confident that the field in its entirety is solving some of the delivery challenges. We have not disclosed gene targets per se outside of the Stargardt ABCA4 gene, but they broadly fall into areas similar to what Mike said earlier in terms of large genes with very high mutational variants. There are targets where in the same molecular reaction, we want to eliminate the toxic species and restore wild type. And we've made great progress there. And then, of course, as mentioned, some targets where you really want to control the expression, those targets that have a very narrow therapeutic index. And they fall broadly into other areas of ophthalmology. As you could imagine, we'd want to continue to leverage what we've built there. But we think there's a lot of application and unmet medical needs in the neurological space. In fact, earlier this year, we announced a collaboration with Roche on a set of neurological targets. So we're excited about that collaboration. We're also pursuing some of our own neurological and neuromuscular targets and a few other programs across different gene areas. And, maybe Mike, you can get into this a little bit, but, there are aspects where Ascidian thinks about our pipeline in terms of what do we want to advance to the clinic ourselves? And where do we think that some programs really make sense to partner with, organizations like Roche who have established certain infrastructure and have, various capabilities that are not necessarily something that Ascidian wants to build today.
That makes a lot of sense. Yeah, it's great. I don't know, Mike, if you wanted to add anything to that, or.
Well, I think that pretty much covers it. Over time, I anticipate that we'll see advances in a variety of different delivery technologies. As Bob mentioned earlier on, our focus and our strategy is to concentrate on making this highly differentiated payload and RNA exon editors. And I think that our pipeline will become a reflection of where the current state of technology is on delivery in different ways. And we think that there'll be a convergence there across a variety of disease targets, cell types, tissue types.
Yeah, I think that's true for every other modality out there. If we're honest with ourselves, so delivery always, I think, remains the biggest, hurdle out there for everyone. So if you were to look through a crystal ball into the future, how do you envision the future of RNA exon editing in the treatment of genetic diseases, let's say 5, 10 years from now? And do you see a role for exon editing in personalized medicine, for example?
That's a big question. So
it is. We'll take it in chunks.
I tell you that I think we have the opportunity to see is some therapies that have, say, 5 years from now, have shown real signs of radical efficacy in genetic diseases that have been very difficult to address at this point. We'd certainly like to think that our lead program in Stargardt's disease would be in that category, that ACDN01 could make a real difference in that quite common genetic blinding disorder and beyond for that. That's what I'd like to see. And I think this is going to happen across a variety of different modalities, if you will. And in some of the glimpses that we see out there, and this is not RNA exon editing, but I was very much influenced by my experience in a previous life. I worked at Biogen and was kind of part of the approval and launch of Spinraza or nusinersen in spinal muscular atrophy. And I think that was a sea change in kind of genetic therapies, particularly in neurological disease way. We see the kinds of exciting efficacy that you can get in areas like transthyretin amyloidosis with different technologies, antisense or RNAis, and I'd say even just today, I think there was a release on CRISPR-based approaches for that. And so these are signs of significant efficacy. And I think that's also what we should be striving for and looking to achieve with these new modalities like RNA exon editing. I'm not a real fan of the type of efficacy that requires complex statistics to squint at a p-value and figure out if it's actually doing something. I think in this field where we're targeting the genetic origins of disease, we should demand of these technologies and the programs that biotech companies are developing that they show very significant signs of efficacy. And that's what we should be striving for.
So you're raising the bar for everyone right now, which is great. I think this is very exciting.
Yeah, we should. I mean, I really think that we should, because again, we're working very close to the genetic origins of disease. If you understand it well, of course, there's always questions on when do you need to treat to have this sort of impact, et cetera, et cetera. But I think it opens a new moment with multiple modalities where we have the opportunity to expand this very significant types of efficacy into a variety of different disorders.
Yeah, that's great. So given that molecular therapy is the journal of the American Society for Gene and Cell Therapy, and the interests of the society with the scientific community, one of my last questions for both of you is, how are you engaging with the scientific community and other stakeholders to advance the field of RNA exon editing?
Yeah, I mean, I think, well, first off, we are working hard to wrap up a manuscript on the ABCA4 program that will discuss everything from how we screened and identified early lead exon editors, how we then optimized that, the studies we ran in mutant cells and non-human primates and human retinal explants, some of the off-target work that we discussed. So that's coming. Stay tuned for that. And, we have been presenting that at some of the conferences. I think the manuscript is coming, but we've really just been focused on, working with the agency and then executing on the clinical trial. I think the other thing that we're really excited about, we talked about this a little bit, is really trying to understand how RNA structure plays a role in everything we're doing, and the tools to not just predict, but to actually empirically measure much longer RNA structures have been developed and continue to develop over the past few years. And we're starting to use that and have found some really interesting thing that we're hoping to share. Because I do think not only do we want to, obviously, our core mission is to advance medicines to patients, but we want to participate in some of the fundamental understanding of RNA processing and RNA biology. We've got such an incredible scientific team, both wet lab and data science, that, their passion for understanding very complicated molecular biology is just overwhelmingly exciting. And we look forward to sharing a lot of that with the scientific community in the coming years.
What I would add is that I hope that people listening to this podcast get interested in this topic and start working on it. And if you do, come talk to us.
Agreed.
Yeah.
Because there's so much that we still have to learn. And the opportunities to fundamentally rewire RNA at the splicing stage is very exciting. And there's a ton of great biology to be discovered, and it has the potential to convert into real meaningful technology. So if you're listening to this, if you work on RNA, get interested, work on it, give us a call.
Sounds great. I can't agree more. And so with that, I want to thank you both for sharing your insights and experiences. And I encourage our listeners to stay updated with the latest developments from Ascidian Therapeutics. And you can find more information on Ascidian Therapeutics on their website at ascidian-tx.com. And with that, Mike and Rob and Bob, thank you so much. This has been really a great learning experience for me. And I look forward to a lot of, great news from your company. Thank you.
Thanks, Paloma.